Hey guys, so this video is going to go over chapter 7, which is all about the structure of our plasma membrane and its function. So we know that our plasma membrane is going to separate living cells from its surroundings. And our membrane, it's actually 8 nanometers thick. And it is going to it's going to regulate that traffic that comes in and out of the cell. And like all biological membranes, our plasma membrane is also selectively permeable. So that just means that it's going to allow some substances in, it won't allow some in, some will cross more easily than others. It's selective. And that ability that the cell has to discriminate against molecules, ions, and other things, so that ability to discriminate in its chemical exchange with the environment, that is extremely fundamental to life. And it is the plasma membrane and its component molecules that are going to make this selectivity possible. So, our cell membranes, they are fluid mosaics of lipids and proteins. And what this means is that the main macromolecules that we have in our membranes are lipids and proteins, but carbohydrates are actually also very important. And phospholipids, they are the most abundant lipids that are in our membranes, and they are considered amphipathic molecules, right? So they're amphipathic because they have a hydrophilic and a hydrophobic region. And because of this conformation that our membrane has, a lot of its constituents also are going to be amphipathic, having these hydrophobic and hydrophilic regions. So our constituents, our membrane components, like its proteins, they're also amphipathic, and a lot of them are actually going to span the entire membrane. And what this figure is showing is just showing us the structure of our phospholipid. So our phospholipid is going to have a hydrophilic head, and then it's going to have a hydrophobic tail. So the head is going to be water-loving and our tail is going to be water-fearing. And our hydrophilic head, it's going to contain this phosphate group, and that's going to be oriented towards the aqueous environment. Meanwhile, our tails, they are made up of fatty acid chains and they face inward. They don't like the water. And this amphipathicity of our phospholipids is going to allow us to form bilayers, which is a really fundamental structure of our cellular membranes. So before we got to the structure of the membranes that we have now, we went through a whole process of different models. And what we have now is called the fluid mosaic model, and it's just going to describe the cell membrane structure as dynamic. So it's fluid and it has a variety of proteins that are going to be embedded inside of or somehow attached to its bilayer, so it's double layer. And because of that, it's actually going to look like a mosaic. So essentially, models of the membranes, they were developed a long time before we even saw the membranes for the first time. And we actually saw our membrane for the first time in the 1950s using electron microscopes. But in 1915, we were able to isolate the membranes from red blood cells, and we chemically analyzed those red blood cells and found that they were composed of lipids and proteins. And then 10 years later, in 1925, we had two scientists that reasoned that our cell membranes must be phospholipid bilayers, and that those bilayers, they were going to be arranged in such a way that the hydrophobic fatty acid tails were sheltered from the water. Meanwhile, our hydrophilic phosphate groups interact with water. So by 1925, we knew already that our membrane was composed of phospholipids and proteins, and we also understood that the tails faced each other and that the heads faced away. So 10 years after that, we had two different scientists. They were named Hugh Davison and James Danieli, and they proposed something called the sandwich model. And what happens in the sandwich model is that our phospholipid bilayer, so this section, it was going to be between two layers of globular proteins, right? So globular proteins. And the early images that we were able to obtain from electron microscopes, it seemed to support this sandwich model. And until the 1960s, so nearly 30 years later, it was widely accepted that that was the structure of our membrane and of our internal membranes. So after the development of the sandwich model, we had continuous investigations. So our research, it continued, and it actually ended up revealing two different problems. So what we found out was that not all of our membranes are alike. And what this means is that membranes, they have different functions and they have distinct chemical compositions and structures depending what cell they're in, where they are, and all sorts of factors. And the next thing that we learned was that the solubility measurements, so basically we learned that membrane proteins, they are not really soluble in water. And we did this by running different kinds of tests, different kinds of measurements and research. So the research, it included a variety of tests, such as solubility tests, which found out that our membrane proteins, that they were found to have low solubility in water. They did detergent studies, and what they did is that they used these detergents to break up the membranes and solubilize the proteins, which revealed to us that they had this amphipathic nature. And then they used spectroscopic measurements, and it just showed that the membrane protein contained significant amounts of hydrophobic amino acids. So all of these findings from these measurements and these tests that we ran, it led us to the following conclusions. It led us to the idea that our membranes are amphipathic, and what happened in the sandwich model is that our phospholipids, they were thought to form that hydrophobic core of the membrane and the hydrophilic proteins on the both sides of the lipid bilayer. And the next thing is that the measurements, it showed that if membrane proteins were located only on the surface of the membrane, that their hydrophobic regions would be exposed to surrounding water. And that's problematic because hydrophobic regions, we know that they don't like water. We know that they don't interact. They don't mix well with water. So if those hydrophobic regions were exposed to water, those proteins, they were going to be unstable and non-functional in those positions. So what was the solution to these problems? The solution was the idea that we have membrane proteins that are going to be embedded inside of our lipid bilayer, where our hydrophobic regions interact with the hydrophobic interior of that membrane. Here, so this is our hydrophobic region, and then these two are our hydrophilic regions. So our hydrophilic regions, they were interacting with the aqueous environment on both sides, and our hydrophobic regions, they faced each other. And this arrangement, it allowed us to stabilize our proteins, and it allows them for them to function effectively. And that is the current model that we have now. So in this protein, we have this part facing out and this part facing out. These are two hydrophilic portions. Meanwhile, this interior is hydrophobic, and it's going to line up exactly with the hydrophobic tails of our phospholipid bilayer. So what ended up happening is that in 1972, we had these two scientists, and they actually proposed that membrane proteins are going to reside, like we see here, they're going to reside inside of the phospholipid bilayer with their hydrophilic regions that were going to be protruding into the cytosol. And that was what gave rise to the idea of that fluic mosaic model that we have now, that our hydrophilic regions of proteins and phospholipids, that they are in the maximum contact with water that they can be. Meanwhile, our hydrophobic regions are in a completely non-aqueous environment inside of the membrane. And there's actually an experiment that was run on the membrane. It used this really specialized preparation technique. It's called freeze fracture. And what that did was that it split the membrane along the middle of that phospholipid bilayer. And when that freeze fracture preparation is viewed with our electron microscope, the protein particles, we see that they're interspersed in a smooth matrix. And that supported the concept of that fluid mosaic model. And even now, with all of our advancement, this fluid mosaic model, it continues to be refined. So like our membranes, they may be more mosaic than fluid. And what this means is that we may have groups of proteins that are associated in specialized patches that are going to carry out common functions. And our lipids, they're also seeming to appear to form more defined regions. And we also think that the membrane may have more proteins than we previously thought. So regardless of this, our membranes, they are fluid. And our membranes, they are held in place by these relatively weak hydrophobic interactions. And these interactions, they are non-covalent forces in which nonpolar molecules avoid water together. And these hydrophobic interactions, they can actually explain certain things like self-assembly, right? So it can explain self-assembly because we're going to have, like, for example, if that's a straight protein, we're going to have these hydrophobic and hydrophilic regions. Let's just say that our blue dots are our hydrophobic regions and that our pink dots are hydrophilic regions. What's going to end up happening is that we're going to get something like this, all right? Something like that, in which our two previously unconnected hydrophobic regions are going to connect like this. Meanwhile, our hydrophilic regions are going to be still separated. So the fact that we have hydrophobic regions that are attracted to each other, it's going to self-assemble because of those interactions. And it's also fluid because lipids and proteins, they're able to move. So most of the lipids and some proteins, they can drift laterally, right? So they can drift this way or this way in the plane of the membrane, but they very rarely are going to move from one phospholipid bilayer to another. So basically, movement inside of our bilayer is going to be within that specific bilayer. So our adjacent phospholipids, like this one and this one, they're able to switch spots, and they do this at about 10 to the 7th power times per second. And those lateral movements, they are really fast. But then we have large proteins like these, and they can also move within that phospholipid bilayer, but they're going to move more slowly. And we have even some proteins that are going to be attached to our cytoskeleton. And the idea is that we have motor proteins that are going to guide certain proteins to that cytoskeleton. And then we also have proteins that are anchored, and those remain fixed. They never move. They're there forever. So the fluidity of our membrane, it's actually going to be influenced by its components. So we've already established that our membrane is really dynamic and fluid, and that part of its fluidity is due to its components. But we have differences in our membranes, such as the amount of saturated and unsaturated fatty acids that it's made up of. And when we have more fatty acids, more unsaturated fatty acids, it's going to be more fluid. That membrane will be more fluid than a membrane that has more saturated fatty acids. So I want you guys to remember that our fatty acids, they are composed of hydrocarbons. So they are hydrocarbon chains, and they are simple lipids, all right? And that our unsaturated fatty acids, they are able to be more fluid because there's kinks that are found in them. They're double bonded, and those double bonds are going to form kinks that stop. It's going to prevent it from packing tightly. But in our saturated fatty acids, they don't have kinks. They don't have double bonds. So they're going to be straight chains, and they're going to be more solid. So our membrane, it's also going to be influenced by temperature. So as our temperature cools, the membranes are going to switch from a fluid state to a more solid state because the phospholipids, they're going to pack more closely to each other. But membrane fluidity, it's also influenced by cholesterol. So our cholesterol, it's going to be wedged in between our fatty acids like we see here, right? So this is all cholesterol wedged in between our fatty acids. And what happens is that cholesterol, it is a complex lipid. Unlike our fatty acids that are simple lipids, cholesterol is a complex lipid. And it is also a steroid. And cholesterol, it does play this really important role in membrane fluidity. So in cooler temperatures, our cholesterol is going to maintain fluidity because it's going to prevent tight packing. But as our temperatures increase, as it gets warmer, for example, like 37 degrees Celsius, cholesterol is going to restrain the movement of phospholipids and then reduce its fluidity. And that's why our cholesterol, it's actually kind of known as a fluidity buffer for the membrane because it's able to resist changes in membrane fluidity as temperature changes. And that's kind of what we're seeing here. When we have these high temperatures like we're seeing up here, and we have no cholesterol, our membrane, it's really fluid, so it's too fluid. But when we have cholesterol, like here, wedged in between our fatty acids, it's going to increase the rigidity of our bilayer. So we're seeing the difference that our fatty acid tails, they are more tightly together versus when we have no cholesterol. Look at how sprawled out they are. And then when we are in low temperatures, our membrane is going to become more rigid. And then there's no cholesterol that stops it from being this rigid. So our membrane, look at its hydrophobic region. It's really tightly packed. But when we have cholesterol wedged in between it, it's going to increase the fluidity that we see. And that is why it is known as a fluidity buffer. So membranes are mosaics of structure and function. Basically, we can think of our membrane like a mosaic, or another word, like a collage of different proteins. And those proteins are going to be embedded inside of that fluid matrix of the lipid bilayer. So like, for example, we have more than 50 kinds of proteins that were found in the plasma membranes of our red blood cells. And proteins, it's going to determine most of the membrane's functions. So the proteins that we have inside of our membrane are going to be the determinants of the specific function of that protein. And the plasma membrane and the membranes of a variety of organelles, they're going to have these unique collections of proteins, which goes back to the idea that all membranes are different. They are all unique from each other. And then we actually have two major populations of membrane proteins. We have integral proteins, which are going to be embedded inside of that bilayer, and then we have peripheral proteins that are going to be attached to that membrane surface or even to integral proteins themselves. So our integral proteins, they're going to penetrate the hydrophobic interior of the lipid bilayer, usually completely. So when we see it like this, it's spanning from one end of the bilayer to the other and even out of it, it's going to be considered a transmembrane protein. And most of our integral proteins are going to be transmembrane proteins, but not always. This one right here, it's also an integral protein, but it only goes to half or part of that lipid bilayer. So when we're thinking about our integral proteins, because it's going to be spanning the membrane or spanning part of the membrane, it has to have hydrophobic and hydrophilic regions. And those hydrophobic regions that are embedded inside of that membrane's interior, it's going to contain these non-polar amino acids that are usually going to be coiled into alpha helices. And the hydrophilic regions of these proteins, they're going to come in contact with our aqueous environments. So we actually even have some integral proteins that are going to have this hydrophilic channel that through their center, which is what we're seeing here. And that's going to allow for the passage of hydrophilic substances. So moving on to our peripheral proteins, they are not embedded into the lipid bilayer. Instead, the peripheral proteins, they are loosely bound to the surface and often to integral proteins. So when we're looking at this cytoplasmic side of the bilayer, right? So this is the cytoplasmic side. This is the intracellular side. We're going to see some membrane proteins that are going to be attached to that cytoskeleton. And then on the extracellular side, right, so the extracellular side, we're going to have some membrane proteins that are going to attach to fibers of the extracellular matrix. And these attachments, they're going to combine to give animal cells a stronger framework than the plasma membrane itself could provide. So this is an example of our peripheral protein. It spans only to the hydrophilic region. It does not actually permeate into this hydrophobic region, which is what we're seeing in both of these. So our plasma membrane has 6 main functions, and it includes the transport of specific solutes in and out of our cells. It's going to include enzymatic activity. For example, some of the enzymes are going to catalyze one of like a number of or a series of steps that we're going to find in our metabolic pathways. They are helpful for signal transduction. They're going to relay hormonal messages to the cell. They're really important for cell-to-cell recognition. So it's going to allow other proteins to attach two adjacent cells together. And cell-to-cell recognition, it's the ability of a cell to distinguish one type of neighboring cell from another. So cell recognition is the ability to distinguish one cell from another. And cell-to-cell recognition, it is completely necessary to the functioning of an organism, because our cell-to-cell recognition, it's important in sorting and organizing cells into tissues and organs during development. It's even the basis of rejection of foreign cells by our immune system. Our cells, they can recognize other cells by binding to surface molecules, which often are going to contain carbohydrates. And so it's going to recognize other cells by binding to the surface molecules that are on the extracellular surface of the plasma membrane. So this is, again, it's so important because, for example, with HIV, human immunodeficiency virus, it's going to infect immune system cells by binding to cell surface proteins. So HIV, it's going to be able to evade the proteins that we find on the surface of a cell. So another function of the plasma membrane is intercellular joining of adjacent cells with gap or tight junctions. And the last function is going to be cell shape, structure, and maintenance. So the attachment to the cytoskeleton, the attachment to that extracellular matrix protein, is going to maintain cell shape and stabilize the location of certain membrane proteins. So our membranes, they are not symmetrical, right? So the inside and outside faces of our membranes, they are different. So they have different lipid composition. And every single protein in the membrane is going to have a directional orientation within the membrane. And the asymmetrical arrangement of our proteins, of our lipids and their associated carbohydrates inside of this plasma membrane, it's going to be determined as the membrane is being built by our endoplasmic reticulum and our Golgi apparatus. So membrane lipids and proteins, they're going to be synthesized in our endoplasmic reticulum, and then we're going to have carbohydrates added to form glycoproteins. And our Golgi apparatus is going to produce glycolipids. So our transmembrane proteins, our membrane glycolipids, and our secretory proteins, they're going to be transported in vesicles to the plasma membrane. And when that vesicle, when it fuses with the plasma membrane, it's going to release secretory proteins from the cell, and the outside layer of the vesicle is going to become continuous with that cytoplasmic, so the inner layer of our plasma membrane. And it's important that we know that molecules that originate from the endoplasmic reticulum's inner face, it's going to end up on the outer face of the membrane. So when we're looking at this leftmost figure, it's just going to demonstrate to us how asymmetry is established. So when we're looking at figure A, what we see is these enzymes called scramblases. They're going to, they're in the endoplasmic reticulum, and they're going to facilitate that even distribution of our phospholipids between the two leaflets of our membrane, right? And then when we're looking at figure B, what we see is that we have another enzyme. It's called flippase, and it's in the Golgi apparatus. And flippase is going to selectively transport specific lipids to that cytosolic leaflet. So right here, it's going to transfer it to that cytosolic leaflet, and that's going to actually reinforce that membrane's asymmetry. So it's going to reinforce that membrane's asymmetric nature. So the role of the endoplasmic reticulum and the role of the Golgi apparatus in lipid and protein processing is that our ER, it's going to synthesize lipids and protein, our lipids and our proteins. And some of these, they're going to undergo something called glycolysation. And that is just the addition of carbohydrates to form glycoproteins. And our Golgi apparatus is going to further modify these molecules, and it's going to produce glycolipids. And those glycolipids, they're going to be what contributes to that membrane asymmetry. So the rightmost figure, this figure here, it shows the vesicle transport from Golgi to the plasma membrane. So basically what happens is that when vesicles, so our vesicles, that came from our Golgi, such as these, they're going to make their way to the plasma membrane. And so it makes their way to the luminal side, which is the side that was originally the inner face of the endoplasmic reticulum and Golgi, which is going to become the extracellular face. Meanwhile, that cytoplasmic face is going to remain unchanged. So these vesicles are going to make their way to the plasma membrane. It's going to fuse with the plasma membrane, and these contents that were inside of the vesicles, they're going to be taken out into the membrane, and it's going to be found on the outside of the membrane. It's going to be found on the extracellular face of the membrane, not on the cytoplasmic face, which is where they originated from. So due to the processes that we have shown in these figures, our membrane lipid and protein composition, it's going to differ between those inner and outer layers of the membrane protein, of the plasma membrane. And this asymmetry, it's really important for functions such as cellular signaling. This is going to function in cellular signaling. It's going to function in immune recognition. And its interactions with the extracellular environment. So our membrane structure results in selective permeability. So our biological membranes, they're going to provide an example of supramolecular structures with many molecules being ordered into higher levels of organization. So these supramolecular structures, they're going to have emergent properties beyond those of individual molecules. And the fluid mosaic model, it's going to help explain how our membranes are able to regulate molecular traffic. So we have this steady movement of small molecules and ions across our plasma membrane in both directions. So for example, we have sugars, amino acids, and whatever other nutrients that are going to enter a muscle cell, for example, and then we're going to have metabolic processes that are going to leave, metabolic wastes products that are going to leave the cell also. So our muscle cell, it's going to take in oxygen and it's going to expel carbon dioxide. But the muscle cell, it's also able to regulate the concentration of inorganic ions like sodium, potassium, and calcium. And it's going to shuttle them one way or another across the membrane, either in or out, depending on the needs of the cell. So our membranes, they're really highly selectively permeable. Their substances, they're only going to allow in some substances, some molecules, they discriminate. And the cell is able to take up this many varieties of small molecules and ions and exclude others. So substances, they're going to move through the membrane at different rates. And the movement of a molecule through a membrane, it depends on the interaction of the molecule with the hydrophobic interior of the membrane. So for example, when we have nonpolar molecules like our carbon dioxide, like our oxygen and our hydrocarbons, they're going to be hydrophobic and they can dissolve easily in the lipid bilayer, and then they can cross easily, and they don't need the help of membrane proteins to do that. But that hydrophobic interior of the membrane, it's going to stop that direct passage of ions and molecules that are polar, which are hydrophilic. And those polar molecules, which include sugars like glucose and even water, it's going to have an extremely small polar molecule that is going to cross that lipid bilayer slowly. And then we have, for example, an ion. So whether it's a charged atom or a molecule and its surrounding shell of water, it can also have difficulty penetrating those hydrophobic interiors of the membranes. So we have proteins that are going to assist and regulate the transport of our polar molecules and our ions in and out of the cells. And our cell membranes, they are permeable to specific ions and a variety of polar molecules that can avoid contact with that lipid bilayer by passing through these transport proteins that are going to span the membrane. And some transport proteins, our channel proteins, they are going to have this hydrophilic channel in which certain molecules or ions, they can use it like a tunnel through the membrane, right? So this hydrophilic tunnel. And then we also have something called aquaporins. And aquaporins, they are a type of channel protein that is going to allow for the passage of water through the membrane. And these aquaporins, they allow for the entry of billions of water molecules per second. And those water molecules, they're going to actually pass single file through its central channel, and it can fit 10 at a time. So it can fit 10 H2O molecules. And by doing this, it's going to allow for 3 billion to pass per second. So think about the fact that if we didn't have aquaporins, we would only have this really small fraction of water molecules that could pass through the same area of the cell membrane in a second. So that channel protein, these aquaporins, they greatly increase the rate of water movement. So we have other transport proteins that are called carrier proteins, and these carrier proteins, they bind to molecules and shape. They bind to molecules, and they're going to change their shape to shuttle the molecules across the membrane. And each transport protein is going to be really specific for the substance or even a group of substances that it's able to translocate. For example, the glucose transport protein that we have in the liver is going to carry glucose into the cell, but it does not transport fructose. And if you remember, fructose is a structural isomer of glucose, but it's really selective in that way. And that glucose transporter, it causes glucose to pass through the membrane 50,000 times as fast as it would diffuse if it had to diffuse on its own. So when we're looking at the figure, that's what we're seeing. We have this molecule enter, and it's going to shape this carrier protein, and then it's going to change, what we see here, down to allow that molecule out and into the cytoplasm of the cell. So passive transport is diffusion of a substance across a membrane with no energy investment. So molecules, they are always constantly moving. And because of that, they have thermal energy, they have heat. And a result of thermal energy is something called diffusion. And diffusion, it's the movement of molecules of any substance to and the way that it spreads out in available space. So diffusion is the natural spreading of molecules in available space. And the movements of molecules, of our individual molecules, they are random. So when we have these three molecules, they're kind of separated and they move in random locations. But when we have a population of molecules like these, when we have a group of molecules, they're going to move in a directional manner. And that's what we're seeing when we're looking at this figure. We're seeing that separate molecules, they're moving around randomly. But as soon as we have this population of molecules, they move directionally in the same to the same place. And that's going to be based on concentration. And it's important that we remember that in a population, we still are going to have random movement from some molecules, but the collective and overall behavior of them is what's going to result in net movement and lead to diffusion. So when we're talking about diffusion across a permeable membrane, we can imagine that there is a permeable membrane that has these microscopic pores that are permeable to the molecules that we find in the solution. So let's just say that this right here is our membrane. And what we're seeing is that we have a bunch of molecules right around here. There's all these molecules in our extracellular space. And the cell, it wants these molecules to go inside of it. So what happens is that each molecule, it's going to wander randomly, but the net movement of all of the molecules is going to be across the membrane, which is what we're seeing here. We're seeing certain molecules start to move down into the intracellular space. And then when that net movement of molecules across that membrane continues, it's going to reach something called equilibrium. And after equilibrium, we get dynamic equilibrium. And dynamic equilibrium just means that we're going to have the passage of these molecules in and out, keeping in order to maintain the equilibrium that we have in this space. So it isn't enough to reach equilibrium for a moment. We have to stay in equilibrium. So when we are void, when we are absent of other forces and a substance diffuses from the area of where it is more concentrated to where it is less concentrated, it's going to be going down its concentration gradient. So the key properties of diffusion, they're fairly simple. What they say is that our molecules, they're going to move down their concentration gradient. So each substance, it diffuses down its own concentration gradient independent of the concentration gradients of other substances. So if we have molecule A that is going to diffuse in this solution, it can diffuse faster than molecule B. So the other important thing of diffusion is that it does not require energy. There is no energy investment needed. Diffusion is spontaneous. So the diffusion of a substance, the diffusion of a substance across a biological membrane, it's known as passive transport, right? So this is passive transport. And it's known as passive transport because it doesn't require energy from the cell to make it happen. The concentration gradient itself is going to represent the potential energy, and it's going to drive diffusion. And because our membranes are selectively permeable, the interactions of the molecules with the membrane, they're going to play a role in the diffusion rate. So osmosis is the passive transport of water. So if we can imagine that we have two different sugar solutions, and they have different concentrations in these beakers, and we can say that these beakers, they're going to be separated by a membrane, which is going to be this line. And that separation is going to allow water to pass, but not sugar. How do we think this is going to affect water concentration? So the water that has less sugar molecules in it, it's going to move to the compartment that has more sugar. It is going to dilute the compartment with more sugar until the concentration of sugar is equal on both sides. And this is what we call osmosis. Osmosis, it is the diffusion of water across a selectively permeable membrane. And osmosis, it states that it's going to move from a lower solute, water is going to move from a lower solute concentration to a higher solute until equilibrium is reached. So both solute concentration and the permeability of the membrane is going to affect something called tonicity. And tonicity is the ability of a surrounding solution to cause a cell to gain or lose water. And tonicity, it's going to depend partially on the concentration of the solutes that cannot cross the membrane, and those are called non-penetrating solutes. And it's going to be relative to the concentration of the solutes that we find inside of the cell itself. So if we have more non-penetrating solutes in the surrounding solution, water is going to tend to leave the cell and vice versa. So we have 3 different types of tonicity. So we have, so for example, if we have a cell that doesn't have a cell wall, like an animal cell, and it's immersed in an environment that is isotonic to the cell, that means that there's going to be no net movement of water across the plasma membrane. What goes in goes out at exactly the same rate. So there's no net movement of water, and that cell, it's going to stay in its normal shape. But if we have a cell that is immersed in a hypertonic solution, hypertonic relative to the cell, what's going to happen is that the cell, it's going to lose water rapidly, and the cell is going to shrivel, and it may even die. And an example of this is an increase in the salinity of a lake. That can kill our aquatic animals, because if the lake water becomes too salty, if it becomes too hypertonic for the cells of those animals, its cells are going to shrivel and die. So we can think of hypertonic as containing non-penetrating solutes. But if we have a cell that is immersed in a hypotonic solution relative to the cell, water is going to enter that cell really fast. That cell is going to swell and it's going to burst. And the process of cellular bursting is called lysis. So in a hypotonic solution, water is going to enter the cell faster than it can leave. And that is also fatal to the cell. So again, talking about our organisms that don't have the rigid cell walls that plant cells have, we're talking about animal cells. And our animal cells, they have osmotic problems whenever they're put inside of a hypertonic or hypotonic solution. So whenever our animal cells are not in a neutral environment, they have a water balance problem. So, when a cell lives in its isotonic solution, it does not experience any osmotic stress because it's in an isotonic surrounding, right? So no osmotic stress when in an isotonic solution. But for example, seawater is isotonic to a lot of marine invertebrates, and the cells of most terrestrial organisms and animals, they're going to be bathed in extracellular fluid that is isotonic to the cells. So if we were to put a terrestrial animal inside of seawater, their cells would swell and they would burst, they would lyse. And but also it follows the concept of when we are in water for too long, why do we get wrinkly fingers? We swell up, and that's because water is entering our cells faster than it can leave. So we have adaptations that are going to help us with osmoregulation because we don't have cell walls to protect us. So for animals and organisms, without these cell walls that are living in solutions that are hypertonic or hypotonic relative to them, they have these osmoregulatory adaptations that are going to control water balance. And an example of this is going to be paramecium. And if you remember, paramecium, it is a protist. It's a freshwater protist. And Paramecium is actually hypertonic to the pond water that it lives in. And despite a plasma membrane that is less permeable to water than other cells, water is actually going to continually enter the paramecium cell. So to solve around this problem, those paramecium cells, they created this specialized organelle that is called a contractile vacuole. And these contractile vacuoles, they function like bilge pumps and they force water out of the cell, which is going to prevent lysis. But we even see osmoregulation in plant cells with cell walls. So we have the cells of plants, some protists, not all, only some of them, these cells of fungi and of prokaryotes, they're surrounded by cell walls. And when we have a plant cell that is in a solution that is hypotonic, what's going to happen is that the cell content, it's going to swell, right? So we have here the swelling of the cellular content, which is what we're seeing, like this and like this. And what occurs is that the elastic cell wall, it's going to exert something called turgor pressure on the cell that is going to oppose further water intake. So once we have this swelling of our contents, no more water can come in because this turgor pressure that we see, it stops water from coming in. And when we have turgor pressure, that's going to make that cell really turgid, right? So that just means that the cell is firm. And that is the healthy state for most plant cells. And these turgid cells, they're actually going to contribute to the mechanical support of the plant. But if a plant cell, if it's in an isotonic surrounding, that means that there's no movement, no net movement of water into the cell. And what happens is that the cell is going to become flaccid, so it's going to become limp, and then the plant will wilt. But when we are talking about a hypertonic solution, the cell wall, it provides no advantages in hypertonic solutions. And this is because as that plant cell loses water, its volume shrinks. And eventually we get something called plasmolysis, which is usually lethal. And what plasmolysis is that the plasma membrane separates from that cell wall. So we have a lot of polar molecules and ions that are normally impeded by the lipid bilayer of the membrane, diffuse passively with the help of transport proteins. So normally we have certain molecules, such as polar ones and ions, that normally cannot pass through the membrane, but they can on their own, so they cannot pass through the membrane on their own, but they can passively diffuse with the help of transport proteins that are going to span the membrane. And that passive movement of molecules down a concentration gradient with the help of transport proteins, that is what we call facilitated diffusion. So most transport proteins, they're really specific and they're going to transport some substances but not others. And we have two types of transport proteins that we're going to talk about. And they include our channel proteins and carrier proteins. So channel proteins, which is what we see here, they have these hydrophobic tunnels, these hydrophobic corridors that our molecules can move through, specific ones. So for example, our water channel proteins, they are known as aquaporins, and they are going to really greatly facilitate the diffusion of water. And we're going to see these aquaporins a lot in our kidneys. And that's what allows our kidneys to uptake water from urine before it's been excreted. So we also have carrier proteins, and what happens in our carrier proteins is that when a molecule enters, it's going to change the shape of that protein, and when that protein shape changes, it's going to allow for that molecule to exit. So carrier proteins, they require the binding to release that transported molecule, because binding is what's going to trigger the shape to change. So we also have gated channels and other transport mechanisms. So for example, we have ion channels that are going to function as gated channels. And these channels, they are going to open or close depending on the presence or the absence of certain stimulus, which includes electrical, chemical, and physical stimulus. So, for example, if we have a chemical gated channel, the stimulus is going to be a substance other than the one that has to be transported. So what I mean by that is that we have chemical A. We have chemical A inside of the gated channel. And then we have another chemical called chemical B. Chemical B is going to activate the channel. And when that channel is activated, it's going to allow for chemical A to enter the cell or exit the cell. So we have some transport proteins that they don't provide channels, but they do appear to translocate the solute binding site and the solute across the membrane as that transport protein changes shape. And these shape changes, they might be triggered by the binding and the release of the transported molecule. So active transport is another form of transport in and out of our membrane, but it's going to require energy in the form of ATP. So it's going to require energy to move those solutes against their gradients. So remember, when we're going with the concentration gradient, it doesn't require energy. But when we're going against that concentration gradient, we need energy in order for that to happen. So some transport proteins, they move their solutes across membranes against these concentration gradients. So it's going to move from the side that is less concentrated to the side that is more concentrated. And these transport proteins that move these solutes, they are going to all be carrier proteins, okay? So these are all carrier proteins instead of channel proteins. And active transport, it requires the cell to use metabolic energy to enable the cell to maintain internal concentrations of small molecules that would otherwise diffuse across the membrane. So when we're comparing, when compared with its surrounding, we have an animal cell. And our animal cells, they actually have a higher concentration of potassium ions and lower concentration of sodium ions when we compare it to its surroundings. And the plasma membrane, it helps to maintain these really steep gradients by pumping out sodium and pumping potassium into the cell. And ATP is going to be the energy that is supplied for most active transport. And the way that it works is by transferring its terminal phosphate group directly to the transport protein. And an example of this is going to be the sodium-potassium pump. So our sodium ion pumps, they're going to generate voltage across our membranes. And what we know is that all cells, they maintain voltage across their plasma membranes. And we can define voltage as the electrical potential that is created by that separation of charges. And so we know that our cytoplasm, that it has a negative charge when we're talking about it in respect to extracellular fluid. And it has this negative charge because there's going to be an unequal distribution of cations and anions on the two sides of the membrane. And the voltage across a membrane, that's going to be known as our membrane potential. And the inside of the cell is negative compared to the outside. So when we have two forces that are combined, collectively, they're going to be called the electrochemical gradient. And the electrochemical gradient, it's going to drive ion movement across a membrane. And then we have something called electrogenic pumps, which they are special transport proteins that generate the voltage gradient across a membrane. So when we're looking at this figure, it's a figure of a sodium-potassium pump. And the sodium-potassium pump, it's this major electrogenic pump that we have in animals that restores the electrochemical gradient. So restores electrochemical gradient. Not only because it's going to actively transport sodium and potassium, but which is going to set up a concentration gradient, but also because it pumps 2 potassium in for every 3 sodium that move out. And that is what sets up our voltage across our membrane. So 2 potassium in for 3 sodium out. And when we're talking about our plants, our fungi, and our bacteria, they have a proton pump, which is that major electrogenic pump, and it's actively going to be transporting protons out of the cell and transferring positive charge from the cytoplasm to that extracellular solution. And by doing that, it's also going to generate voltage across our membranes. And these electrogenic pumps, they help store energy that can be tapped for cellular work. So the first thing that we see when we're looking at this figure is ion movement. We're seeing our sodium ions being transported out of the cell, right? So our sodium ions are starting here in our intracellular space, and they're going up this protein, and then it's leaving the cell into the extracellular matrix. And we're also seeing that potassium, it's being pumped in. Potassium is these yellow ones. Yeah, this is what we're seeing. We have potassium that's being pumped in. So potassium and sodium, they are actively being transported. And this active transport, it requires ATP. And here's our little ATP. So ATP, it's going to be hydrolyzed into ADP and an inorganic phosphate, which is going to fuel our ion transport. And because that pump is moving out 3 sodium ions out and 2 potassium ions in, it's creating this overall net negative charge inside of the cell. So we see here. And this gradient, it's going to just further drive this diffusion of ions, which is really important for things like nerve signaling, for cellular homeostasis, and even for muscle contraction. So when we're looking at the right side color gradient, it's indicating that the relative distribution of our potassium and our sodium. And it's just highlighting the difference in concentrations that we see across the membrane. So we have something called co-transport, and it is a single ATP powered pump that is going to transport specific solute. So it is a single ATP powered pump that transports a specific solute indirectly, and it's going to drive the active transport of several other solutes in that mechanism called co-transport. So as the solute that has been actively transported, as it diffuses back passively through a transport protein, its movement, it can be coupled with the active transport of a different substance against its concentration or electrochemical gradient. And we actually see this commonly in our plants. But being able to understand our co-transport proteins, having the ability to understand water balance in animal cells and osmosis, it's really helped scientists and researchers develop these effective treatments for things like dehydration that can result from diarrhea, which is actually a really serious problem in countries that are developing that have prevalence of intestinal parasites. And what they do is that they're going to give patients a solution, and that solution, it's a drink that contains this high concentration of glucose and salt. And then those solutes, they're going to be taken up by our co-transport proteins on our intestinal cell surface, and then it's going to pass through the cells into the blood. And that resulting increase of solute concentration of the blood, it causes a flow of water from the intestine through the intestinal cells into the blood and rehydrates the patient. So when we're looking at this figure, what we see is that we have this proton pump right here at the bottom, and that proton pump is going to use ATP to actively transport these hydrogen ions out of the cell. So this is extracellular, and this is intracellular. And because we are transporting these hydrogen ions into the extracellular matrix, this is going to create that electrochemical gradient, and then we're going to have more concentration of hydrogen ions outside and then a more negative charge inside of the cell. So positive out here and negative inside of the cell. So what happens is that we're going to have this hydrogen sucrose co-transporter up here, and this co-transporter, it's going to allow these hydrogen ions to diffuse back into the cell passively down their gradient. And as these hydrogens move back in, sucrose also is going to be transported into the cell against its concentration gradient. And this process, it indirectly relies on ATP as the proton pump maintains that hydrogen ion gradient that powers co-transport. So we have something called bulk transport across the plasma membrane, and it can occur by either endocytosis or exocytosis. So when we have small solutes in water that are going to enter or leave our cell through the lipid bilayer or through transport proteins, so we're going to have these solutes. They're going to enter and exit the cell through the lipid bilayer or through our transport proteins. We're also going to have particles and large molecules like polysaccharides or proteins that are going to cross our membrane in vesicles. And like active transport, these processes are going to require energy, which is also ATP. And when we're talking about exocytosis in specific, what happens is that we have a transport vesicle, right? So here we have a vesicle, and that vesicle, it came from the Golgi apparatus, and it's moved by the cytoskeleton to the plasma membrane. And when the two membranes, so the membrane of the vesicle and the plasma membrane, when they come in contact, the bilayers, they fuse, and then eventually it's going to spill out the contents to the outside of the cell. And we see a lot of secretory cells use exocytosis to export their products, like our pancreatic cells that are going to secrete insulin into the blood by exocytosis. So exocytosis, basically, it occurs when a cell releases substances by fusing a vesicle with the cell membrane to move materials out of the cell. And endocytosis, on the other hand, is the reversal of exocytosis, but it uses different proteins. So during endocytosis, what we have is molecules like the ones that we see here, and a cell is going to bring in these molecules and particulate matter by forming new vesicles from the plasma membrane. So here we have this small area of the plasma membrane that starts to sink down. It starts to form like an inward pocket, and that pocket is going to get deeper and deeper, which is what we're seeing, and eventually it becomes its own separate vesicle. And that is how the material gets inside of the cell. So if we can think of it, endocytosis is the process of a cell taking in substances, forming a vesicle from the cell membrane, and then bringing those materials into the cell. And we also have 3 different types of endocytosis. We have phagocytosis, which we can think of as cellular eating. We have pinocytosis, which is like cellular drinking, and then we have receptor-mediated endocytosis. And it's important that we remember that our endocytosis requires ATP. It requires energy. So, receptor-mediated endocytosis, it's going to allow the cell to acquire these like bulk quantities of specific materials that can be that they may be in low concentrations in the environment. And this is really important to humans because our human cells, they're going to use this process to take in cholesterol for use in the synthesis of membranes and as precursors for synthesis of steroids. And cholesterol is going to travel in the blood in the form of low density lipoproteins, protein complexes, and lipids. And these lipoproteins, they're going to act as ligands that are going to bind to those low density lipoprotein receptors that are on the membrane, and then it's going to enter that cell by endocytosis, which is what we see. We see these molecules, we see this pocket forming, and eventually we get this separate vesicle. And vesicles, not only do they transport substances between the cell and its surrounding, but it provides a mechanism for rejuvenating or remodeling our plasma protein. So we have endocytosis and exocytosis. They're going to occur continually in most eukaryotic cells, but the amount of plasma membrane in a non-growing cell is going to remain fairly constant.